Okay, so welcome to everybody to this new installment of the ILS webinars. I'm very happy to be here with Professor Giller, Ken Giller. He's emeritus professor at Wageningen University in the Netherlands, and he has a long career. He's focused on sub-Saharan Africa and soil problem linked to soil fertility. And today's webinar is on the topic of the legacy of the project N2Africa. So without further ado, Ken, please, the microphone is yours.
Okay, well, thanks very much. I'll just work out to share my screen. If I can. Okay, is this working? Can you just confirm, Nelson?
Yeah, sure. I see your screen,
great.
full screen.
Well, thanks so much for the invitation to talk to you today, and great to see some familiar names in the list as people were joining just now. Yeah, and what I want to do today is to talk about this project N2Africa, which took up a good 10 years or more of my own career, and of many people, in fact, and to look back a bit on the legacy of that project in particular. So I think you all know there are different types of legumes and different legumes used in agriculture. So here in this slide, just some illustrations on the legume green manure. So we've got mucuna, crotalaria. We have legume forages. This is Stylosanthes in Madagascar. On the right here, we have, on my right, the legume tree fallows here in Zambia. And in the center there, the grain legumes. So different types of legumes that can be used in agriculture. Nitrogen fixation, for those of you who don't study the topic, it's easy to demonstrate in the laboratory here in the glass house. Some yellow plants you can see which don't have a nitrogen fixing rhizobium strain compared with those which are fixing nitrogen actively, dark green and prolific in contrast. That's more difficult to demonstrate in the field, but this is an experiment from work we were doing in northeast Thailand quite some time ago, where we have some genotypes here in the front. These are yellow, they're nitrogen deficient. They're actually varieties of groundnut which don't have the ability to form nodules. So they're nitrogen deficient. In the middle of the plots there with groundnut fixing nitrogen. And of course, these non-nodulating varieties aren't much good for farmers, but they're great for us as scientists because they allow us to make accurate measurements of the contribution of nitrogen fixation in the field. Zooming in on the groundnut root system, you can see these roots of groundnut are covered in these round bumps, these nodules. And if we slice through one of those roots, we have the root here in transverse section, the nodule on groundnuts, and you can see the center of the nodule is full of these very densely stained cells. They're packed with bacteroids, the modified bacteria, which is where the process of nitrogen fixation takes place. Now, going straight to N2Africa then, this was a project which I led from Wageningen University together with IITA, International Institute of Tropical Agriculture, ILRI, AGRA, many national partners, and we worked in 11 countries in Africa, Ghana, Nigeria, Ethiopia, Uganda, Tanzania, but also these other countries. So 11 in total. Our first phase of the project was actually $22 million, where we looked at what we called a proof of concept. And then we were, a second phase, we had $30 million scaling through partnerships and institutionalization. So $52 million in total, all funded by the Bill and Melinda Gates Foundation. And what I can say is that we obviously reached a lot of farmers through this. It sounds like a huge amount of money, but when we're 11 countries, 10 years, we had about 30 partners in each country. You can understand that we spread the money actually quite thinly. So the approach we used then to increasing inputs from nitrogen fixation was, first of all, trying to get more legumes into the farming systems using a technology targeting approach. We were looking at increasing legume productivity, so particularly good agronomy and phosphorus fertilizer being important there. We were trying to select the best legume varieties, selecting rhizobium strains and using inoculation, and then linking to markets and creating new opportunities for farmers to increase the demand of legumes, because obviously if you're going to produce more legumes, we need somewhere that then to be sold. So we worked on different grain legumes, particularly cowpea, groundnut, soybean in West Africa. In East and Central Africa, we included common bean, chickpea, and faba bean, particularly in chickpea and faba bean are cool season legumes grown in the highlands of Ethiopia. And in Southern Africa, common bean, groundnut, soybean, and cowpea. But just to make the point, we didn't focus on green manures, cover crops, and fertilizer trees at all, because our experience is that the primary goal of smallholder farmers is to produce for food and for sale, with soil fertility improvement. Although it's an important role of grain legumes, it's really very much a secondary goal rather than their primary goal. So we focused purely in this project on the grain legumes. And we use this approach, which I think all of you would recognize as scientists working with legumes, a genotype by environment by management approach, but where the genotype, of course, is the interaction between the legume genotype and the rhizobium strain. These are then targeted to different environments, and I already mentioned the targeting of different grain legumes to different regions. And then, of course, management being then the inoculation, fertilizer, seeding rates, plant density, good agronomy, basically, to make the system work in the field. And we called the project a development to research project. So we started, if you like, with rolling out technologies, and I'll talk more about this. So taking the best technologies that we had at hand, rolling those out with farmers, so dissemination development as the core, then with a strong monitoring and evaluation shell here, which improved our learning and on the performance of things in the field. That took us into research to actually understand what was working and what wasn't working, to actually create these feedback loops to improve productivity when we encountered problems in the field. And I'll give an example of that as we move forward. So we started with what we called these very simple tryouts, farmer tryouts, single replicate experiments, if you like. And here's an example: soybean with no inputs, soybean with phosphorus, soybean with inoculation, and soybean with both phosphorus and inoculation. But on fairly large plots, 10 by 10 meter plots, so farmers could really see the benefits of the technology. And these were both then demonstrations of the technology, but also they were farmer experiments, participatory experiments, if you like. So inoculation, here's an example of two different inoculant products, one legume fix from the UK, one biofix from Kenya. So inoculation is a very simple technology. Here we have a demonstration in the field. This lady looking rather suspicious, where we add the inoculum into the bucket as a demo for farmers. That's then simply shaken very gently with a lid on it. And here we have inoculated seed ready for sowing into the soil in the field. So it's a simple technology to apply for farmers, for smallholders. And these are the sorts of results that we achieved. In the picture here, you can see a control plot in the foreground. This is in northern Nigeria. And behind the farmer, we have the plants with phosphorus and with inoculation, which are clearly doing very well indeed. So this is one of these 10 by 10 meter for treatment experiments. So in this graph, we've got experiments from northern Nigeria over a couple of years, 2011, 2012. And you can see that the mean effects go from a control yield of 960 kilos per hectare here, almost doubling with phosphorus and inoculation, with both phosphorus and inoculation giving a substantial effect. But actually, we found that this is an additive effect of adding phosphorus and inoculation together. And you can see that in terms of the spread of points as well. The ones here at the top are where we've got single superphosphate and inoculum compared to only inoculum on its own or just single superphosphate on its own. But what we've done here is plotted the grain yield in the control plot on the bottom axis, and on the upper axis here we've got the yields with phosphorus and or inoculation. And you can see there's this massive spread of yields from around 2 tons per hectare in good fields to very, very poor yields in some fields. And this then, of course, becomes a topic for research. Here we are in Ethiopia, so an experiment here with common bean, a similar type of experiment. In the foreground here we've got as a demonstration 2 different varieties of beans with no inputs, no rhizobium, no phosphorus. In the background, the improvements in yield then from adding both rhizobium and phosphorus together. So really very substantial effects also here in common bean. And similar to the trials that I showed you earlier, using these farmer tryouts, the control yield plotted on the x-axis and the treatment yields on the y-axis here with common bean. And again, we're going from a control yield here of 1,400 kilos per hectare up to more than 2 tons per hectare with both phosphorus and inoculation. And again, we find no direct interaction between P and I. These are additive effects. And in total, during the time of N2Africa, we had a huge number, nearly 100,000 of these farmer tryouts located across all of the different target countries. We had able to sample about 4,500 of those in terms of getting yields. We had another 4,000 demonstration trials, some 645 on-farm research trials, which were more detailed. And our overall effects then, if you compare researcher yields to farmer yields, showed definitely 50% yield increases in the technology plots compared with farmer plots. And again, looking at these incremental yields in researcher plots. And I say these are mean effects, which include, of course, many fields where the effects weren't so significant. When it works, it's fantastic. Here with an extension worker in northern Nigeria, in his right hand, on the left here, you can see the soybean plants without phosphorus inoculation. In his left hand, on our right, you can see then the fantastic yields with both phosphorus and inoculation together. And farmers were incredibly enthusiastic, particularly about inoculum, because inoculum is very cheap. Relatively, we can cost it at less than $5 per hectare, and gave really very significant effects. In northern Ghana, farmers used to talk of inoculant as this black magic powder. But going back to this graph, which I pointed out before, we had these huge problems in some areas of very, very small yields and no response to fertilizer. If we zoom in on one of these fields, this is in northern Ghana, you can see a very poor establishment. With when we go into the field, everybody's saying, look, it's been weeded late, and you can see behind there's weeds here just been taken out. So that was probably the reason. But then where the farmer's standing, there's this brilliant looking plant. Zooming in, you can see one plant is doing really, really well. And it's actually due to problems of soil fertility. This was the lucky plant that probably received a gift of a passing cow in the previous season, where some manure had fallen into the field and actually gave them the benefit of soil fertility. So we sampled these soils, taking them into these double pot experiments where we can do missing nutrient experiments. Here we're Samson Foley doing these experiments in a northern Nigeria in Kano, where we can look at then which are the missing nutrients which are actually causing the problems. And in this case, actually, it was a whole cocktail of different problems, including sulfur and micronutrient problems. So in those poor soils, in fact, even in the control here, you can see these, we think these are manganese granules, so real problems of the soils, problems with no micronutrients, clear magnesium deficiency and phosphorus deficiency. And that actually allows us then, through these feedback loops, to go back to think about having a different type of fertilizer blend to address these problem soils. And working with Yara, we actually ended up releasing a new blend of fertilizer called Yara New Legume Fertilizer together with the company to address these problems. So that's one example. But we had also other simpler approaches, sometimes here in Malawi, where farmers grow groundnuts on a single row of plants on a ridge simply by using better varieties and doubling up the sowing density. These varieties resistant to rosette virus, we could get much better yields, almost doubling yields. And these are varieties with disease resistance. So simply through double row planting, farmers were very enthusiastic to adopt these technologies. And we've seen these taking off in Malawi in the past years. In Rwanda, we have climbing beans, again on the left, climbing beans with no manure added, on the right with manure and phosphorus, and on very small farms, these climbing beans are wonderful to intensify production vertically, where you can get the beans up to 3 meters tall, where I, as a reasonably tall person, feel completely dwarfed by the crop. And these beans producing up to 5 tons of grain per hectare. This lady is Mrs. Gasilida, and this is actually a variety which was developed during participatory breeding, which is named after her. After the crop of climbing beans, if we compare here plots with maize after maize or maize after climbing beans, you can see a very clear residual effect in terms of the soil fertility benefits of the legume to the subsequent crop. And I can't present all of these results, but we had a special issue, Open Access in Agriculture Ecosystems Environment in 2019 with 12 papers actually summarizing the results of the project. And I'll, as I move forward, simply highlight some of the main points without actually showing you much in the way of data. But I want to particularly focus on what we've done since the end of N2Africa. But during N2Africa, we used different approaches to meeting farmers, to getting legumes out to farmers. We had a big focus on what we call public-private partnerships. So here's one in Southeast Ethiopia, where the researchers at one side here were working with different extension agents, with a company who was actually providing seed, with an inoculant company from Addis Ababa who was providing the inoculant, and then with a commodity supplier who were doing trainings on grading of grain for the market. And working in these areas with agro arrangements, we could work with up to 23,000 farmers to grow these Kabuli seeded varieties for export. But working through these public-private partnerships gave us a wonderful scaling model for some of the best technologies. Here we are in the field with Charlene McCoyne from our colleague from the Gates Foundation at the time. And these were these large Kabuli varieties that were being grown there by farmers groups for both sale in Addis, but also actually as an export crop. In other countries where companies weren't so active, we had big partnerships with different NGOs. This is one women's group in northern Uganda where we were working with World Vision, had a very strong gender focus, working with women farmers. So looking at building capacity and cooperation, focusing on storage, collective marketing, and diversification away from pure systems with pure staple crops. And here's one of the youngest members of the women's cooperative here presenting me in the field with a chicken as a gift for the benefits of the project. It's not something I brought back on the plane with me, I have to say, but it was something of the appreciation of the people when we were there in the field. And I'll come back to this example later as how we've been able to work with them to scale up these activities with soybean in northern Uganda. In West Africa, one of the technologies we work with cowpea were these short duration varieties. They're varieties with which are white seeded. Many cowpeas in West Africa are grey or brown seeded, but these are good ones for the market. They're short season, which means that they yield during the hunger period when it's very green, but when there's been no food production since the year before. This allows 2 crops in the season. And these are very nutritious. Legumes, of course, providing energy, protein, but also particularly minerals and beta vitamins. And actually, that led to us in 2013 being awarded this prize for N2Africa by the Secure Nutrition Network, the Harvesting Nutrition Prize. Here we are at the bottom here as one of the 3 winners because of the way that we could show that our demonstration and working with women's groups was actually leading to changes in consumption, which could actually then benefit the nutrition of the household. In Tanzania, we worked with a whole range of organizations in this, what was called in Swahili, the Mharai Bingwa project, the Champion Bean Project, bean thinking. So here we are with N2Africa ensuring a consistent message, working with comics and other ways of reaching people through radio. So particularly with Farm Radio International, putting out messages to farmers, but having a back office where people could text in their questions, and particularly where we could do surveys with farmers to ask farmers what were the technologies that they were looking for. And then from this research that we were doing on what the farmers were looking for, we could actually then link up with the Soil Health Consortium and the African Fertilizer Association to actually make sure that the right inputs were present in the agro dealers and that the agro dealers were also then trained in the proper use of technologies. So if you like, creating a whole alliance around bean production to stimulate the uptake and use of inputs. And one thing that we really learned in this, that there were many other organizations. So we were here as N2Africa, but there were many other organizations, Legume Alliance, the Soil Health Consortium, another USAID project, another project here with a whole range of partners in which N2Africa worked as well. And they were all putting out ideas of how beans should be grown. But when we actually asked them what the messages should be for the radio, we came up with different ideas. And we were able then, through this working with the radio station, we actually had to come out with a consolidated approach, which actually helped the projects to learn from each other. And then to link up with these value chain products, which we're actually then looking at the delivery at the other side. So we found that these working through these alliances, such as this Legume Alliance that I just mentioned, were very useful in getting joint learning among different partners and different projects. One of the things we learned was one of the most popular bean varieties was actually this variety Soya Njano. We were surprised when we heard farmers in northern Tanzania were growing soy, but it turns out it wasn't soya bean. It's actually a round-seeded variety of common bean called Soya Njano, means yellow bean, which was actually renowned for its fast cooking and had a very good price in the market, you can see here, compared to other beans. So this one and this Kablangeti were actually beans with fast cooking times. And those are ones that we could then feed back into research, because when we started, this yellow bean wasn't actually being included in the breeding programs, but then we could actually feed that back to research, again through these feedback loops. We worked with researchers and research facilities in different countries to help to equip laboratories and greenhouses. And we worked with many PhD and research students. Here, Amaral with Mariangela Hungria, one of our advisors who won the World Food Prize this year. Amaral did his PhD in Brazil with Mariangela from Mozambique. And here, George Mwenda, who is doing actually competition studies with these beautifully stained rhizobium strains on nodules using different gene markers. And another student here, Ashinafi Gunabo, worked extensively in Ethiopia. Just a few examples looking at distribution of rhizobia, the genetic studies of the different rhizobium strain performance, looking at phylogeography of also rhizobia for chickpea from Ethiopia, and then actually exploring those symbiotic interactions. So a whole range of different trainings of people. And again, I'll mention Ashinafi as we move forward. But one of the biggest problems that we faced was actually getting the inputs to farmers. And this remains to be a huge problem in Africa. We worked on seed delivery systems through community-based seed systems in many places, but also linking to seed companies. Inoculants were registered for sale in all of the countries. When we started, there were no requirements for registration, no systems for registration in the countries where we were working. Locum production was scaled up in 4 different countries, and particularly in Nigeria through this product Nodumax, which was produced through the IETA as a new company, which remains in production. And then working also with importers such as Rhizobacter from Argentina and Legume Technology from the UK. But one of the issues that we found is that many of the local inoculants, and the names are blanked out here, were actually highly variable in quality. Here we've got dilution series on agar plates, and you can see these local inoculants containing many contaminant bacteria. Now, these inoculants work. They work if they're used quite soon after manufacture, but they tend to have a very short shelf life and low numbers of bacteria, compared with inoculants such as this one from Legume Technology, which produces on a sterile carrier, so it only contains rhizobia in the inoculant. It can deliver up to 1,000 times as many rhizobia per gram of inoculant. And that means that you get consistent performance and stronger responses. And this sort of technology is what Nodumax has now been trying to adopt, using a sterile carrier rather than using these local carriers, which are simply pasteurized. Because as you move through a value chain in Africa, you need a product which can actually last well on the shelf. These local inoculants really have a shelf life of less than 3 months, so it gives you very little time from production to delivery. And Bruce Knight here at Legume Technology, he's got a grant now actually where he's producing he's got a new machine which should be able to produce up to 5,000 packs a day, with the hope that these machines will then be deployed to African countries in future. And these packets, of course, are very suitable for smallholders, these small packs, compared to the big packs which are then used on larger commercial farms. Now here's our research roadmap. So moving forward, we need then to think further about breeding for nitrogen fixation. We recognize that farmer preferences are driven largely by taste, cooking time, and market opportunities, and continuous breeding is needed, particularly for pests and diseases. We need to understand competition more. So competition with local strains is the key to success for inoculation, and particularly need to ensure farmers can access high quality inoculants. So further work on rhizobium strain screening, particularly for legumes like common bean, chickpea, groundnut, and cowpea. We understand then very often our scientists are telling us that we have negative nitrogen balances with our legumes, but we actually find very strong residual effect of the legumes, which are much larger than due to nitrogen alone. So the control of pests and diseases, and particularly things like the parasitic weeds like striga. We need then to address these problems of poor and depleted soils, so multiple deficiencies of nutrients need these multi-nutrient blends, which tend not to be available in many African countries. And then, of course, we need to be tailoring technologies for this huge diversity of African farming systems and smallholder farms, and that needs this iterative participatory experimentation and modeling in order to scale out the promising technologies. One example we had was in DRC, in the Congo, in South Kivu. We actually did seed diffusion studies where we found that farmers who received a new variety shared with at least 5 other farmers, which indicated that our reach was much, much larger than just the farmers that we were directly interacting with. And again, working through these public-private partnerships was a strong approach. Now, I want to finish off with a few examples, though, of what we've been doing since the end of N2Africa in 2019. And particularly, that's been trying to look at other ways of moving legumes into the field and also getting good policy support for sustainable intensification with legumes. Now, one is then looking here at soybean. And if you look at soybean production over the years, you can see a very disappointing case in terms of Africa, with soybean increasing exponentially in the world. But if we zoom in on Africa, we actually see an exponential increase in Africa. It's just in terms of global production, it's still very, very small. And urbanization is driving a massive demand for soybean as a feed, particularly for poultry and fish production. Yet Africa's importing 80% of its soybean. So there's a huge opportunity there for smallholders and for the substitution of imports. Here in northern Ghana, we've continued with a project, the Sustainable Soybean Initiative. In 2023, I was there with these groups who were looking then at demonstrations, again with women's farmers. And we found in that season, in particularly in this growing season in Tamale in northern Ghana, when this was during the crisis of the start of the Ukraine crisis, when fertilizer prices shot through the roof and farmers couldn't get fertilizers. Farmers actually went over to put 60% of their land under soybean, because on the right here you can see that's what maize looks like without fertilizer, and on the left, of course, soybean doing very well without fertilizer. So farmers recognizing very much the benefits of legumes when they couldn't access nitrogen fertilizers. In Malawi, visiting again a couple of years ago, in 2023, nearly 3 years ago, we found actually the landscape covered in soybeans. These farmers here threshing soybeans at the house, and soybeans then being taken off to the market. So these local traders, actually, this lady was coming along with a small bag of 3 kilos of soybean. She put it on the scale, she received some kwacha, she went off to the market to buy food for the evening. So these traders then consolidating the soybean into sacks, going off to the growth points. These growth points actually going to these processing factories, which are actually now processing a huge amount of soybean in Malawi, where we've seen then production take off here up to now to somewhere close to 300,000 tons of soybean being produced. And I can remember back in the day when we were doing demonstration with soybean, and it was hardly a known crop in Malawi. But here it is actually replacing tobacco in these areas where tobacco is collapsing as a cash crop, soybean coming in as a cash crop, which is then being processed for export and for particularly for poultry feed and of course for soybean oil. So here we have soybean oil actually on the shelves in the supermarket in Malawi. Here we've got these soybean products being sold as sort of fast food in the supermarket. And here's Frederick Kawalewale, who we helped to actually privatize inoculants in Malawi. In his country, his company, Agricultural Input Suppliers Limited, is still going ahead with producing inoculants in Malawi as a spin-over from the time of N2Africa. So we've really seen then these massive changes in production of soybean in different countries. And I'm not saying it's all down to N2Africa. We've definitely played a role in that transition. But how do we actually get then inputs to farmers? We had one PhD thesis, Koneti Ayisiga, who worked with N2Africa. She continued to work on with these information communication technology platforms in northern Uganda, where these platforms working through mobile phone systems could help to actually be a broker between different institutions, NGOs, research, production suppliers, extension agents, what we call village-based agents, to actually link then into farmers and to output markets. And she found that actually providing loyalty incentives, and in this case, providing simply tarpaulins to farmers from the input suppliers, actually increase the farmer's loyalty to the buyer. And at the same time, it actually, through increasing that loyalty, it increased the use of inputs and actually led to increase in uptake of different varieties and improving soybean yields. So these information technology systems have a very strong role to play. Another, I think, very exciting example from Ethiopia was looking at the bundling of inputs and services. So for farmers, if they were offered seed, rhizobium, inoculant, and fertilizer together as a bundle, we actually found it only increased uptake to 29% of farmers compared to 26% of farmers who are buying the inputs individually. And I have to say, this was an experiment with over 300 farmers who were actually using their own money actually in the shop. It wasn't actually a giveaway at all. We actually added a microloan because we thought, well, maybe it's microfinance, but that actually didn't actually increase production at all. Actually, it went down from 29% to 26% by adding a microloan to the bundle. But when we added a market contract, then up to 60% of the farmers bought the bundle when it was bundled with a market contract. And then putting in both microloans and the market contract increased the uptake to 80%. And I think this shows very clearly then, if we can put together this bundle of inputs together with market contracts and give farmers some security of the market, we can actually boost the uptake of inputs and boost productivity and farmers' incomes very strongly. It's very much their uncertainty of what's going to happen if they invest in terms of market, which often precludes them from actually taking up these inputs. And then my final example then is working in southern Tanzania. This is the grain basket of Tanzania, again through digital technologies, this management-based advisory for farmers, a survey-based tool, which records farmer practices for soy and maize fields, and then actually uses that information on practices to actually generate advice, field-specific advice for farmers. Starting off with the tool, and this is work of my colleague Jens Anderson and his team working there. He found the first thing that farmers didn't know was actually their field areas. So building in a GPS measuring tool actually allowed them then to actually apply inputs much more regularly. And through recording farmers' practices, we can see what farmers are doing and the problems they're facing in terms of pests and diseases. And then to generate these different advices, which are field specific for the different farmers. Some of the learnings from that are that farmers are using very little fertilizer and manure on soil directly, and that currently inoculants are still hardly available. And here, farmers often relying on their own stored seed, and these are germination rates depending on how many years that seed's been recycled. So recycled seed for soybean is reasonable for the first couple of years, but tails off over time, showing that we really need to introduce new seed into the system. And here the seed problems. Now, this can help enormously then in moving forward in terms of the types of information and inputs that we need to actually try and get to farmers. And through this initiative, the Sustainable Soybean Initiative, working with partners in the Southern Highlands, been able to register over 20,000 farmers to roll out these activities. And different types of advice coming through. Here, this one on crop rotation with soybeans, and particularly this is work of Mukoma Kila Kila for a PhD. And I just zoom in to show you here that if we're looking at the second season maize production, maize after maize with no fertilizer, 1.3 tons, maize after maize, which was fertilized in the first year, and that's a carryover effect, 2 tons. But soybean and maize with no fertilizer in yield one, here giving us 3 tons. So again, the benefits of soybean in rotation actually is a way of getting nutrients into those systems, which then the soybean can benefit from as well. So really taking some of these things to scale. Final example, I was in Ethiopia in October. Here's Ashanafi Ganabu, who set up his laboratory, and he's continuing now as Dean of Research and with his testing of rhizobium strains here on soybean and other crops in his glasshouse in Arba Minch in southern Ethiopia. And he's got projects now then trying to roll out here mung bean and other legumes in southern Ethiopia in areas where it's almost 100% of the land is under cereals. So again, a legacy here through our capacity development of Ashanafi and his generation of students who are taking this work forward. So my last slide, well, it's quite clear that we need the good legume genotype, the good rhizobium targeted to the environment, but in terms of productivity, it's actually management, agronomy, phosphorus, which overrides. We need nitrogen fixing grain legumes which are flexible. We can find different fits which fit all types of farmers, both rich and poor. So different types of technologies for different types of farmers. We focused in N2Africa on this idea of continuous learning and improvement, and we continue to carry on learning, of course. There is a key role of strategic public and private partners in the project. I think we built a great network and collaboration across Africa, which is being carried forward by the many different researchers in the different countries. So we've definitely been putting legume nitrogen fixation to work in African agriculture. Not to say that there's a lot more to do. And reaching small farmers in different places is all about context, and particularly finding ways of getting those inputs to farmers that they need in order to boost their yields. And I finish then with this link to our website, which is still there. There's lots of materials there. And thanks to all of the many scientists and partners who are involved in this project, who, of course, were responsible for making all of this work and carry on that work now into the future. And at that point, I hope I've left you about 15 minutes for discussion. So thanks very much.
Thanks, Professor Giller. It was a very nice presentation. You touched a lot of interesting topics, and it was impressive, a very impressive project.
Thank you.
So I see that we have already a question in the chat, and then if anybody in the audience wants to ask question, just pick up or write in the chat, and I will read it back to Ken. So This question goes like that. A great presentation. Can you say more about negative nutrient balances of bean plots? Does this only happen to beans that are not inoculated?
Yeah, in my own time in research, as with others, no doubt, we've done these calculations of both partial and full nutrient balances of legumes. And very often we come up with this idea that there's a negative nutrient balance. But I think these are actually very misleading in different ways. One is that we tend not to include the below ground contributions from legumes. And we know that's not a huge amount of nitrogen, but there is nitrogen in the roots and in the nodules. But in particular, we tend not to capture a lot of nitrogen, which actually, certainly in some legumes such as soybean, actually falls back to the soil in leaf before harvest. So particularly where you've got a closed canopy, you get self-mulching, and there's quite a lot of nitrogen going into the soil from leaves there. So although we come out with this idea that the legumes are taking away more than they fix, I really wonder whether that's true. Definitely, the calculations look better when we have inoculation and the legumes are growing very prolifically. That's definitely true. But I do think that our negative nutrient balance is usually misleading, because when we grow crops after legumes, compared with after cereals, we find very large impacts in terms of better growth of the subsequent crop. And particularly, we find that that's also proportional to the biomass in the legume very often. So I think it's actually as much to do with calculation errors as that actually lead us to misleading conclusions. But it's a difficult area, actually. But thanks for the question.
Thanks. I hope that answered the question. Otherwise, you can write on the chat. There's another couple of questions. So, this one, please comment on the inoculants from Argentina and UK compared to locally developed inoculants in terms of effectiveness and availability.
So, yeah, I mean, I really need to make this very clear that these locally produced inoculants, if they're taken from the factory, and if we bought them and we use them in our experiments, we could show that they worked. But the big problem is in Africa, your supply chains are difficult. Yeah, access is difficult at getting things out to smallholders who are distributed in villages all over the place. So to be able to only produce for 3 months, if you have a 3 months to 6 month shelf life, means that you're so restricted in how much you can produce and get it out through those supply chains. Now, the benefits of inoculants like the ones from legume technology are then that you have a sterile inoculant carrier. You have only the strain there. Now, if that inoculant then gets exposed to high temperatures, the numbers might go down, but if you put it back at room temperatures, the numbers will come back up again. So that it actually you can have a shelf life of up to 2 years. And you really need that long shelf life in order to be able to get the numbers of rhizobia onto the seed so that it really works in the field. If these poorer quality inoculants are stored badly, they tend to lose their viability because the contaminants overgrow the peat. And when we've recovered these inoculants from smallholder suppliers, from local agro dealers, we can often find no rhizobia in them at all because they've lost that effectiveness. So that's why I really think that we've got to have this focus very much on the very best quality products, because that can give this consistency of response, and then farmers won't be disappointed, basically. I see some people have their hands up as well, Nelson.
There's a few people with their hands, so we go back. The first hand is by Stefano.
Yeah, hi. Well, thank you for the very nice and interesting presentation, first of all. I guess I have a bit of a broad question. I'm wondering if this approach that you use on farm research in a very large, extensive scale could be something that we can implement also in a more industrialized agriculture, such as in Europe, where no doubt we have an increasing interest in legumes, but still there are some hinders in the adoption by farmers. And do you have some thoughts on this?
Well, sure. I mean, what we were challenged, of course, in the Gates Foundation wanted, and our donors, they wanted to make sure that we were not only doing research, they wanted to see things moving into the field. And so what we adopted was really an applied research approach, though, where we focused on this idea of learning loops. So we take the best technology we have, we test it out, we see where does it work, and then when it fails, then the question is what do we then do to improve it? Yeah. So I think that these ideas of participatory research, I think, which are adopted very much in Europe now, working together with farmers to develop technologies, I think that's something that we learned very quickly in the tropics and we brought back to Europe in many ways, because in the tropics we were often confronted with systems that we didn't fully understand. We needed the farmers' knowledge. And I think this whole idea of sort of technology starts in the lab, the scientists in their white coats go into the field and then tell farmers what to do. I think we've really got to escape from that. I think we need to be in this collaborative mode of really working together with farmers to develop technologies. And on the point of legumes in Europe, I mean, I think the reason that legumes don't go so fast in Europe is simply that the economics are so badly stacked against legume production in terms of the yields and the prices farmers get for their products. And that unless we do something really serious about that, I don't see that changing so easily. Yeah? We can carry on saying we need to grow more legumes, but unless it makes economic sense for farmers, they're really not going to do it. Yeah. So I think it needs some very strong policy address. My own motto is, for everything, answer to everything is eat more beans. But of course, not everybody necessarily agrees.
Thank you.
Okay, my connection went off for a moment. On that, I did notice that during the presentation, mainly there were many references to using for the legumes for poultry, for oil. Maybe I lost something. There was not much focus on direct consumption.
Sure. I mean, I did mention the cowpea variety, and this we won this prize for harvesting nutrition, and of course, for common bean, very much the common bean is a widely consumed legume. I mentioned cooking times as being a main point there. But it's true that quite a lot of our work has been driven around these opportunities for soybean, because soybean production is taking off so rapidly, driven, if you like, by the urban markets, by the demand for poultry, and then alongside that, aquaculture, yeah, particularly production of catfish in West Africa is a massive consumer of poultry feed, of animal feed, soybean cake, most of it imported from Argentina, which could be being produced by the smallholders in country. Yeah.
Okay. Thanks. I see that Bowini, I'm sorry, I don't know how to pronounce your name, Bowini. You both raised your hand, and you wrote in the chat, so you can just
Yeah,
pick
I
up.
also posted it, but thank you very much. Hi Ken, thank you for the presentation on N2Africa. Great to see its work as it was N2Africa and now as it is continuing. I have a broad question, which mainly what key steps would you see in improving input availability for farmers, especially smallholder farmers, maybe especially focusing on seeds?
Okay. I mean, this on the seed side, and Bowie, yeah, very nice to see you. So haven't been in touch for quite some time, but I recognise your name very much. Um, on the seed side, I mean, it really is differentiated for the different types of legumes. For soybean, because it's a much more, if you like, commercial crop, and because its seed is very short-lived, I mean, you really need to replenish seed nearly every year, then it's commercial seed producers are basically the way to go. And we see certainly in countries like in Zambia, smallholders buying seed directly from companies. When it comes to cowpea, to beans and the like, then um, they don't have the same interest from commercial seed producers. So we tend to focus very much on community-based seed production. So helping farmers to produce what we call quality declared seed. So instead of them just buying grain, which often contains disease seed and the like, actually helping farmers to learn how best um, to grade and to keep their seed from year to year. I mean, we have this wonderful benefit of many of the legumes are self-pollinating, very, very little outcrossing, so they stay true to type over generations. So that's a real benefit for farmers. And when we introduce new varieties, we definitely see this idea that if varieties are popular, they tend to be spread through the local communities from farmer to farmer. On other inputs, purchased inputs, the biggest problem in many ways is fertilizer. I think inoculants can be very cheap if we can get them moving. That's a problem in itself, but particularly the fertilizer prices are so high in Africa compared with other parts of the world that it makes agriculture very unprofitable in many ways for farmers to invest in. And certainly with what's going on in the world now in terms of these appalling conflicts and the like, it's only really going to make things much, much worse in Africa in the next years.
Okay. If Bowini, if you are satisfied with the answer, we move to Barbara, and also you also wrote in the chat, so you can read your own question.
Yes, hi Ken. It was
Barbara.
good to see you, and it was a great talk, and I think really a project with a huge impact. It's really amazing. Thank you so much for that. So I was wondering your micronutrients besides phosphate. So how much did they boost the yield increase by inoculants? Like if you have a bad field with these spots and magnesium was missing and so on, and you compare this bad field plus inoculants with your micronutrients added plus inoculants. So did it give a much higher boost of yield after inoculation?
Sure. So I find it really hard to differentiate, if you like, between because it's always gone in these multi-nutrient blends, if you like, but it's definitely the case that we have situations where just adding phosphorus actually can cause more problems in these very, very poor soils because it locks up the remaining zinc. Yeah, so zinc has been one of the micronutrients we've identified as being a problem. I would say that overall, I think that micronutrient problems are fairly locally restricted, where they can be locally very important. But I don't think as a whole we should ever walk away with the idea that we need to be applying micronutrient blends everywhere. I think they need to be targeted to the specific environments where we really identify the problems, because of course it adds extra complexity to getting nutrients into the system. But definitely we see in some places magnesium, calcium being problems, some places sulfur. Of course, Africa has very old soils, so we need to think then locally about what the local problems are. But particularly in light sandy soils, we see some of these problems popping up. But I couldn't tell you whether there's any direct interaction with the inoculant there, Barbara. Anyway, but thanks and nice to see you.
Another small question: the best formulation for the delivery of the inoculants, what did you use there? Was the most robust and best one peat, or do you have other formulations which were suitable for the smallholders?
that of my experience, and I know that in Brazil, Mariangela would be saying, I mean, liquid inoculants have really taken over, but there you're dealing with situations where you can have cold supply chains where liquid inoculants are going to be
Exactly.
okay. I think in Africa, liquid inoculants, it would be the wrong way to go. I think you're unlikely to get survival through to the farm. So we've relied very much on the, if you like, the old traditional way of having well prepared peats, but with which are modified with nutrients in order to give you the best growth in the peat. Yeah. So
And are those
and
recipes on your website?
no, no, not directly. I mean, obviously, I mean, people like Legume Technology, they don't divulge directly their formulations, but but I can give you some tips if you want. I
I will
mean,
contact you later.
essentially, yeah, I mean, but basically, finely ground peat together with a little bit of nutrients. Um, Many people also use these polymers, polyvinyl pyrrolidine, which actually just increase the adhesion to the seed. So I think that there are some little tricks there to make them extra effective in terms of carrying the numbers of rhizobia onto the seed. Yeah.
Okay. Thank you.
Cheers.
I think this answers also two other questions that we have seen that I can read in the chat. There is somebody that asking, can you talk about advances as well in the issues about formulating fertilizers? More or less you answered already, but if you have something explicit on the advances.
Well, I mean, my own feeling is, I mean, we work with different fertilizer suppliers. I mean, one of the big problems in Africa is nearly everywhere you go, all you can buy is diammonium phosphate and urea, and they're the cheap ones, And I think ideally what we'd have would be, particularly for these more problem soils, is multi-nutrient blends with calcium, sulfur, maybe some zinc and everything, which would be more tailored to specific crops. But of course, they come at an extra price, and if you're dealing in a system where production is so price sensitive, it really becomes an issue. So I don't have I think we know what types of nutrient blends we need for different crops. We could definitely come up with better nutrient blends for legumes, but they would tend to be more expensive, and therefore farmers often resort to using diammonium phosphate because it's the cheapest form of phosphorus. So it's not the scientific ideal, but it makes most sense definitely for
From
the
the
farm.
economical point of view, the practical point of view. Yeah, there was a follow-up question here in the chat from the same.
I see Navdeep has a hand up as well.
Yeah, just to close this, the follow-up question was about soil mapping, soil info system to have locally adapted formulations.
Yeah, I mean, there's a huge amount of work gone on to soil mapping in Africa. So we have maps which they claim have got 30-meter distance coverage. My understanding, though, is that what goes on in terms of crop production in a field is as much to do with what happened in that field the year before as it does in terms of the overall soil characteristics. And therefore, I think we can get away without a huge amount of new soil mapping. I mean, we know where the big problems are. My investment would be on getting the inputs to farmers. And I think, these approaches of bundling and the like are really exciting in that line. And of course, we can refine things all the way in terms of breeding, in terms of strain selection. And we can carry on doing our science in the background as well to continually improve the technologies. But we've definitely got technologies that work really well that we should be pushing out as fast as possible. Yeah.
Okay. Navdeep, thanks for the patience. It's your turn now. I see your hand up.
Yeah, thank you. Thank you, Ken, for the nice presentation. And I'm Navdeep. I'm a PhD student in University of Guelph in Canada. So my question is very simple question in terms of farmer side. So what is the practical inoculant rate you have applied for the soybeans or common beans, like the results you presented before? And second of all, how much phosphorus do you think those crops need?
Yeah.
And yeah, go ahead. Sorry, I'll ask the third question again.
So, I mean, the phosphorus one's easy to answer. I mean, we usually find, I mean, about the smallest amount you can easily apply is around 20 to 30 kilos of P per hectare, and that's what we go with always. Never more than that. That's enough just to get the crops going. And if that would be applied more regularly, then that'll have additive benefits over time. What was your question on the rhizobia? Sorry, on the inoculants?
The inoculants, I just want to know like how much like inoculants farmers need to apply maybe per kg of the soybean seeds or common bean seeds. I don't know, maybe like 2 mils, 3 mils, I don't know.
Well, I mean, it comes in these small packets, and I mean, the point there is I mean, I know at one point I've seen packets made of the sort of tea bag size, but if they get so small, of course, then they're much more susceptible to handling damage. So we don't usually go smaller than a few 100 grams in terms of package size, and that would a package of around 500 grams would be enough for about a hectare, yeah, if it's a good quality inoculant. So, you know, it's you have what, about 60 kilos of seed per hectare of soybean, 50 of other legumes. So, you know, it works out at about that sort. So we think of, you know, costs of around $5, roughly $5 American per hectare, something, which is really a very small amount, of course, in relation to what it can do in terms of boosting yields. Yeah.
Okay, nice. Okay, thank you. But like, I have third question now. So building on Barbara's question about the formulation, have you done like seed inoculation or the liquid application of the inoculant? Maybe it's a weird question.
I mean, you should be talking to the microbiologists around like Barbara about this, but you know, people have put a lot of emphasis in the past on trying to get freeze-dried inoculants, liquid inoculants. I mean, I see these days people talking about putting all sorts of inoculants on seed and then you know, drying it. But usually, the massive death of bacteria occurs from drying if they dry as you're trying to inoculate. So I think these sort of dry formulations where you put it on and then it's dried, you're making things much more difficult because you need to provide some sort of protector to keep the bacteria alive on the seed surface before it gets into the soil. So peat's been brilliant because, you know, as long as you inoculate, you sow directly, you carry the live bacteria into soil in large numbers, and it's really the tried and tested technology. And I'm no expert on inoculant technology, but I've not really seen anything that can beat that so far, I'm afraid.
That's great. Thank you.
There's still a couple of questions from the chat. I will go through them quickly. One is about high soil salinity. So nitrogen fixing strategies to be adapted, especially for abiotic stress, especially for high soil salinity. And then I give you right away the other one is about intercropping with other crops and sunflowers specifically. So this viewer asks, says that in Rwanda, we found that a land equivalent ratio of around 1:4 mixing sunflower and soybean.
Yeah.
I guess I cannot understand if it's 1.4 or 1:4. Can you, in general, can you say so?
Sure.
Topics
well, I mean,
salinity and intercropping.
Yeah, I mean intercropping comes in so many different forms, of course, and you know there's a huge amount of literature published on this. If you do actually, if you look at that special issue that we had in agricultural ecosystem environment, we've got some great papers there on intercropping. So I mean, I think legumes intercropping works really, really well. Soybeans probably the worst one for intercropping of all the legumes. It seems to be the least tolerant of intercropping. Much better grown in open fields. On salinity, we've not done a huge amount on salinity in Africa. I mean, definitely I've worked on salinity and nitrogen fixation a bit in the distant past. I think the legumes themselves are highly sensitive to salinity and don't particularly do well in saline soils. One of the good things is if you inoculate, if the legume's going to grow okay, once the bacteria get inside the nodule, they're isolated from the salinity so that they don't have the problem of surviving, if you like, in direct contact with high salt concentrations. Although we know that high salt concentrations will kill off the living bacteria. I've seen some super work done by the University of Adelaide on selection of forage legumes for very saline problems in Australia, for instance. But I think a lot of that's to do with getting a better adapted legume as much as the nitrogen fixation per se.
Okay. I think, yeah, from the chat they say that it's 1.4 the ratio.
Oh yeah, okay, sure.
Okay.
But I mean, sometimes you can get land equivalent ratios going way above 2 if in very good conditions with legumes. I mean, it's sure.
Okay. Okay.
Super.
Let's see. I think it's that.
I think we should let people go.
We should probably let them go. So the very last thing, obviously, we are going to thank Professor Giller for the very interesting presentation. And Professor Giller is in Wageningen University, so if you want to contact him, and you find his profile on our website.
Sure.
And the very last announcement, you still have 4 days to send your abstract for the upcoming conference, which is going to be in June, 8 to 12 June, our conference that is a one every 3 year opportunity. So you better move and send us your abstract. The deadline is in a few days. Okay, so thanks again, Professor. It was
nice to see you all.
Sorry, which conference is that? Sorry, which conference is that?
It's our conference, the International Legume Society Conference. It's one every 3 years. And this time around, we have seen, we talked about a few times, but maybe you missed the messages. This year is in Dubrovnik, so in Croatia. And every 3 years, we meet up, all the legume people, we meet up. And talk about a lot of interesting stuff. The deadline for abstract is on March 15, and early bird registration is somewhere at the end. I should be prepared. I should be more prepared on this. So
April 15.
April 15. Thanks, Tom. April 15. So next month. So you have still a month to enroll for just the conference. Okay.
Thank you so much.
Okay.
Yeah.
So
I put my email address in the chat in case people want to get in touch. So yeah. But thanks very much, Nelson, for the opportunity to talk as well.
Sure. Thanks
My
again.
pleasure.
Thanks to all the participants, and see you all in a month.
Yeah. Cheers then. Yeah. Bye.
Thank you.